we do is before we actually hit these things here, let me just go back and talk about, we had our activity on Friday, and I think actually this was like a really fun activity. In fact, I enjoyed actually doing some of the, I did the activity and I enjoyed it again. Even though I wrote the activity, I still enjoyed doing it again. And I wanted just to go over the answers with you, just so that you have an idea about what you were supposed to have done. And so when we were working with this activity, what we were doing is, remember we were looking at reads, we were looking at all these different chunks of basically code from DNA, which has been kind of like cut up into jigsaw puzzle pieces, and then those jigsaw puzzle pieces have to be reassembled. And hence, that's where the assembly part comes from. And so one of the big points about the sequence assembly is that it's just too much information for computers to take one piece of DNA and go from the very, very, very beginning of the DNA and work their way all the way to the very, very end of the DNA, and to figure out some kind of find out what the sequence is. It just doesn't happen. It's just, your computer is just going to overload. And the thing is, though, that one of the logistics about that, though, it's actually quite difficult to ensure that you can actually, grab the beginning of the DNA and make sure that and get that DNA to actually sequence the DNA from the very beginning. Let me just change my camera here just so I can see what so I'm a little bit not so much in your face. There you go. Maybe that's better. We have this other camera here, which I think provides better resolution so that the videos actually come out a little bit more crisp. Anyway, never mind. So anyway, but getting back to this thing here. So what we do is when you're working with your sequence assembly, your sequence is really going to be you're really going to grab a piece of DNA, you clone it a whole bunch of times, like a bunch of times, and then you cut up all those little clones into these tiny little fragments. And that's where you get this kind of scene here. And then the computer algorithm that you're using is actually taking each of those fragments, and like a jigsaw puzzle, it's figuring out where it goes in the sequence. Now, because you have so much repetition, since we cloned the DNA so many different times, we have all these different chunks. You have like pieces that have overlap, and then you have like, kind of matching pieces, which that have the same overlap, and you have other pieces that have overlap, but you have to figure out where those pieces go. So just because you have overlap doesn't mean that those overlapping pieces go exactly there. In fact, I'm looking at this illustration right now, and I would say that for the sake of example, this is actually quite easy for, this is how we discuss these things, but it's easy for us to learn how this stuff works. But when you just see like 4 overlaps like this, just 4 bases which overlap this, I guess this kind of a reddish colored sequence fragment and this cyan colored sequence fragment, just 4 is really not very much. If you think about it, 4 bases could randomly just show up from mutation over evolutionary time. And so it doesn't really, it's really difficult actually to say that those 4 bases are a good overlap for those 2 chunks of code. And so what I mean by this is that if you are trying to figure out like whether the red piece goes first at exactly this place here, whether maybe it goes down the line someplace and there's another overlap of GTTA, it's very hard to say. And so your algorithm really is looking for like longer chunks of overlap. The longer the better, and the longer the better is necessary because it's less likely that those 4 bases are just randomly the same. Just randomly the same. So if you have like a longer word, maybe it's like 10 to 15 or 10 to 20 bases, then that's more interesting because that suggests that that overlapping region right there is not left to chance. It's not random, but actually that could be a very good argument for that these 2 pieces really are, that they really are overlapping. There's just too much common code. And so for that code to randomly, spontaneously show up that way is just almost, it's almost a 0 probability. And so you're saying that because they share the same code, they have to be overlapping. And so that's really the nature of the algorithms that we're using here. And that's figuring out like, what is the overlap? How do we determine whether we have enough overlap between each of these sequences to say, yeah, this is the right order for these things. But once you finish everything out, you get this assembled sequence. And the assembled sequence is really what they would call the consensus sequence. It's like, this is how we make everything agree, all the fragments that we found. we found fit into this order, and so that is our consensus. So fast forward to this activity, activity 9. What we've done is I've given you some chunks of, this is actually a little demonstration here, but I've given you some chunks of code which have been cut up into little pieces, like chunks of DNA, which have been cut up into little pieces, and it's your job to figure out how they went back together. But you don't do this manually. Instead, you use something called GC Assembler, which is actually a pretty good program. I mean, it's got a huge workload that it has to do, but it does a pretty good job. It's pretty efficient. It gets the job done quite quickly, which is actually quite it's quite nice to find that there's a tool out there that does this. Because if you had to write this tool yourself, it might take you a very long time to invest yourself in the actual algorithm, to implement everything, and then to find the computational power to make it all happen. But it seems like this is a tool that's just sitting online and it has everything ready for you, working, and it's fast. So what you do then is you go ahead and you just dump in your read data. You could dump it into this box here, or you could upload it using your upload FASTA format. But you're going after that file that I gave you with the sum or the activity, and you put it into this form, whether you upload it or just dump the data in there. I would recommend uploading it. And then when you run it, you get this, you get some information. So what I wanted to show you is like, what is the information that this thing gives you? It gives you 2 different sequences. And then you take those sequences, and somewhere in here, it asks you to figure out, here, looking at the text on the results page, is how many sequence reads were rejected. Well, that's something you can see in your contigs file. But what I'm looking at here is that according to BLAST, what is the genome that we've just assembled? And so this is like an excellent use of BLAST right now. And that is that once we have figured out, like, how to put all these reads back together, actually how do we use the reads to make contigs, then put those bigger contigs together back into the sequence. We run that sequence into BLAST and we say, okay, BLAST, here's this unknown sequence. I don't know what it is. Tell me what this thing looks like in terms of comparison. And then it will tell us that I've compared this sequence and I found that this sequence is very similar to other salivarius A genomes, which I have on record. So this is really, again, one of those core research tools that you use in bioinformatics, where you're going to be just comparing sequences. I know that when we first started talking about just comparing sequences, many of you may have thought to yourself, like, well, how is that necessary? How is that good science? Like, how is that actually helpful? Well, the thing is, though, that because we have other versions of this salivarius A genome on, that someone has sequenced, some scientist somewhere has picked up this DNA virus, and they've said, I'm going to sequence this thing. I'm going to leave this virus in the genome or in the database here, and I will use this as a reference point to anything else that comes in. And if anything else that any other sequences that come in appear to look like this virus, then maybe they are the same, or maybe it's a relation. And that's exactly what we find here. That BLAST does show you that this unknown sequence is very, very similar to the salivarius genome, which suggests that it's a relation of the salivarius genome. Now, when you look at how similar this thing was, you should have noticed that, in fact, the numbers that I have and the numbers that you have may be different. But you may notice that the next question here is, did you find that the sequence read that were assembled cover the entire genome, or did the gaps, or were there gaps that remained? And this is, you can look at the numbers if you want, where I said that basically I found the newly assembled gene or contig that I have was 7,982 bases long, while the salivarius A genome is 7,989. These numbers may be different in your particular case. I think this one will be the same, but I think that this number might be different because of updates to the data. It seems like they're always updating, they never stay the same. But what it's saying is that because these numbers are different, if that's your sequence, and this is the other sequence that they found on record, because the bases are different, you're differing here by 7 bases, it's different, it's slightly different. So there's something going on. That means that either your genome has 7 bases missing, or this other genome here has 7 bases more. And so you don't really know who's correct here, but you do know that there's enough information to say that your bases or your sequence that you found is close enough to the salivarius A genome in terms of, in terms of stretches of code which aligned almost perfectly, that it has to be a relation. In other words, BLAST is saying to you that it was so close, it was so kind of similar that it couldn't be anything else. And if you looked at the e-value that you see here, in fact, I think I may have mentioned this someplace, but if you look at the e-value in your BLAST output, that was a number that was very, very close to 0, saying that it was just almost impossible for these two sequences to be so closely related if it was just random. They're just too long. There's too much stuff going on. So you'll see that it must be the same. It must be the same, maybe not the same organism, but a very, very, very close relation to the organism. And so in biological speak, we're saying that actually it's probably the same organism. And so of course you have to do more research and find out whether it really is the same thing. But you're saying that it's so close, you're only 7 bases off. It's probably the same thing, just with some mutations. Maybe it was isolated from some new place. Another thing you could say, though, by the way, is that because it's 7 bases different, that could also be an error that happened back here. Maybe an error happened back here, where maybe 3 bases were knocked off the end on the left, 2 bases were knocked off the, or 4 bases were knocked off the sequence in the front. Who knows? There could have been a whole bunch of little errors that happened. And so, in fact, the data is the same, but it's just that you had an error that happened or that creaked in at the sequencing stage, and so therefore you have the differences in these lengths. It's actually very cool when you see how this is being used in, I mean, big labs, I say big labs, like labs that are working with COVID-19 virus variants are actually doing exactly the same thing that you did for this activity 9. They're doing exactly the same thing. They start here, they figure out what the sequence is of this new variant, and they find out what the or they grab a piece of, or they have a sample of the DNA, they clone it a whole bunch of times, and then they use technology like this to figure out all these little chunks of DNA, what the actual sequences are for the little chunks of DNA. So in other words, they're making reads, and then they start here again, and then they have to assemble those reads into this longer consensus or assembled sequence, and then they run it through something like GC or EG assembly or assembler. They probably use, for some of these more mission critical, I'll call them, types of experiments, they probably do the same thing maybe hundreds of times just to be sure that they're correct. I mean, after all, you have people's lives hanging in the balance here. And so you probably do this experiment several times, but maybe hundreds of times over the years. But you wouldn't just do it once and say, oh yeah, I'm done. But you'd have to do it like many, many times to be sure that this actually is the proper arrangement of your sequence of the virus. The last thing here is the celluvirus. Okay, so here's the celluvirus genome. And I think there's one more question here. And there's one more question that was at the very end that you were to respond to, and it was this question in blue. To see how these parameters affect the assembly, in the enable sequence assembly process options, try setting the overlap percent identity cutoff to 100%. And so when you change it to 100%, did you find any, was there any difference in your, was there any difference at all in your, when you're working through this? Did you, in your result, in terms of your results, did you find anything different? And I don't think you did find anything different. In fact, when I ran this thing through, I'm looking at this, it shouldn't make any difference at all. The percent identity cutoff wouldn't make any change because it's already so close that it counts as the same. So changing the overlap percent identity cutoff to 100% does not change the results. The first resultant contig is still, in my case, it was 7,982 bases and still aligns very well to the celluvirus A genome. The second contig is also the same size and aligns to the same sequence as in question 6. This shows that perfect matches are not required when assembling a genome sequence. This is interesting because a perfect match, that's why you may find that your numbers are slightly different from the numbers that I have up here. And that is because I'm looking at, when I'm running my numbers, the BLAST is picking, it basically forms a giant list of the data that it has available, and then it does its analysis. and then it has another list of the analyses outputs, and then it chooses the very top of the list. And the top of the list for me might be different from the top of the list from you, even though the numbers are still comparable. They're still maybe 7 bases or 6 bases or whatever it may be. But because the data is changing, there's always updates. You might find that your difference is maybe not 7 bases, but maybe 6 bases, or maybe even less. But the result is still the same here. Changing the overhead or the overlap identity cutoff to 100% does not change the results. They're still so close that they're basically the same thing. So I wanted to spend some time just going over this to end the suspense, just so that you have an idea about what you would have seen when you had run this activity. I think that it's actually a very important activity, and it's very cool if you think about it, because this activity is actually the same stuff. It's the same action, the same science that professional bioinformaticians would do. For today's slides, we have genome annotation and sequence-based gene prediction. So it makes sense that after we've been talking about actually working with genome assembly projects like this, you may have realized that a lot of the data we've been looking at so far, when we look at DNA in gene banks and NCBI and all those things, there's information about proteins that exist there, or there's information about how long things are, or information about how long the sequence is, or information about I don't know, the organism where it's come from, or there's some kind of a name, for instance, for this sequence. I mean, how do they know this stuff? I mean, someone just brings you this piece of DNA from the wild. They have no idea where this thing came from. They bring it to you as the head bioinformatician, and they say, "Tell me what this thing is. Where did it come from?" And so your job is to identify this. In other words, what you're really doing is you're annotating your sequence, and that basically means take the piece of DNA and figure out what's going on in the code. In other words, where are the proteins? Where are the genes that make proteins, rather? Where are the other interesting effects that we would expect to see if we're comparing this sequence to another sequence? And so you're really kind of characterizing this. You're identifying your sequence, but you're also characterizing the sequence and saying, this is obviously the cellovirus, or it's something else. And then you have to describe that. But all this information is absolutely essential when you put a piece of DNA into these databases, because other people are going to be using this data for their research. So you need to make kind of predictions. We'll start off with predictions here about where the genes are. And this is the hard part, because someone just brings you a piece of DNA, and you think to yourself, okay, well, I know this DNA is supposed to do something, but what does it do? Like what genes does it hold to make which kinds of proteins? And so you don't know, because it's just a bunch of A's, T's, G's, and C's, and so it's very difficult to find these things. And so you're really kind of doing a study of what the genes, what they look like here. I mean, we're asking ourselves, and basically, in general, are there specific rules or syntax, if you will, that genes must follow in order to be called a gene? Are there behaviors that these genes exhibit when they are a gene? Like if a piece of DNA is a gene, does it behave differently in some kind of experimental situation than a piece of code which is not a gene? In other words, is there a trend? And so we're asking ourselves, what are these features that we could use to determine what a gene does, or what a piece of code would do to become a gene, or to be named a gene that would create a protein? And so we ask ourselves, well, why don't we just run the DNA run the DNA? That's actually kind of a complicated idea. But why don't we just see what proteins come out of this genome and just run it, as a program here, and see what proteins are created? And the reason for that is because if a protein is being created from DNA, it must mean that the DNA contains a gene, right? But the thing is, though, that one of the problems is that there's something called gene silencing, which is exactly what it sounds like, where a gene has basically been stopped, and it doesn't work because it's been turned off. For some reason, the gene is non-functional. The code is there, but it's non-functional. That means it's somehow been, there's no power going to it in some sense. It's like it's not doing what it's not doing anything. And so we have what we call landmarks. Now, these are not my pictures, by the way. I didn't take these, but I'm sure that you could tell me exactly where each one of these photos was taken. All right, I'm thinking that the upper left picture. There's no words next to this, but what would you recommend? What would you think this would be? Probably Paris. It is Paris. Why do we know it's Paris? Actually, that's a good question. Why do we know that this is Paris? There's no sign there. I don't see anything written in here. There's no street signs. In fact, this could be any river or lake or body of water. But there's one piece of information there that we look at, which is in the center of this photo, which is the Eiffel Tower, and that tells us that this is actually in Paris. Anything else could be anywhere else in the world. I mean, this could be some nice place in I don't know, maybe in another country. It could be even in Washington, D.C., who knows? But when you see that Eiffel Tower, though, it tells you one thing, that could not be anywhere else. That has to be Paris. And when we look at, for instance, Big Ben on the right-hand side, and the House of, what was that, Westminster Abbey, that's Parliament. You're looking at, actually that's not Westminster, that's Parliament. But you're looking at the Houses of Parliament, and you're looking at Big Ben, and so that would tell you that has to be London, because there's nowhere else that has such an austere clock like that. Big Ben. Below, here's some other pictures. I don't know if anyone has been to Pittsburgh, but if you've been to Pittsburgh, then you'd recognize those beautiful yellow bridges which extend from one side of the rivers to the other side. I mean, you have to see these bridges when you go there. But again, we're looking at specific pieces, and even this you can look at this what do you call this? I guess a cable car, which is featured in this picture in the lower left, which tells us that that this is another big feature of Pittsburgh. And so this picture could only be in Pittsburgh. This place, there's a place on the right, lower right, some building. Does anyone recognize that place? That's here on campus. That is our own Bentley Hall. And so the idea is that you recognize certain aspects of these photos without having to know everything, but you just look at certain pieces. Your eye goes to certain places, Bentley Hall, or the Eiffel Tower, or the bridges, or wherever it is, and you just know where that was taken. Otherwise, all these pictures have trees in them, except for I guess the one in London. But they all seem to have certain, they all have like a skyline. They all have other things, which makes them beautiful. But it doesn't tell us anything about where this stuff actually came from. Where do these pictures actually come from? So we look at specific types of landmarks. Well, guess what? We can use that same reasoning inside these DNA sequences that we find. And in fact, one of the more interesting landmarks in DNA is something called the Shine-Dalgarno sequence, which is kind of an area that's upstream of of some of the genes. And so, for instance, I'll just read this here. There's these two groups, or there's two researchers. I think they're Australian researchers, but they showed that there is this sequence here, which is ACCU UA, that's RNA, but they found this pyrimidine-rich area, which is really an indication that a gene is coming up. And so as you go through the DNA and you start looking at the code itself, you'll reach this region here, this ACCU UA, or if you look at this, the opposite, or I guess the complementary sequence over here. But if you find this region, then that's a suggestion that there's going to be something coming up here, and you'll see that, for instance, there is a start codon coming up right there. There's your AUG, and that should be, I think, your methionine. So, for instance, when you're looking at, if you look at the RNA, you'll see this, but also you can find this in DNA, because if you know what the RNA is, you can get back to the DNA. And so what I'm trying to say, though, is that you have these landmarks that exist. And actually what this thing is, it's actually kind of interesting, but it's a binding site for where some molecule will stick to the DNA or the RNA rather, and start making things happen to produce this protein. This happens in prokaryotes, and so you have this this area there. But it's actually quite interesting, though. But you have, and I do recommend you have a look at this paper, though, because when this paper came out, it was just really kind of, I hate using the cliche earth-shattering, but it really was something exciting, though, because they're always, in science and bioinformatics research, they're always looking for ways of finding the onset of genes. And the reason for that is because you don't know really what these genes are until perhaps, I mean, before this time, I guess you didn't know that there was a gene until you started noticing that there was a protein being made. And so then you'd start looking around for, well, what could have made that protein? And there's the gene. But with this observation, with the Shine-Delgarno sequence, we're saying that if you find this sequence, then you're going to find a gene. A gene will soon come. The RNA sequence serves to attach the ribosome to the mRNA to begin the protein synthesis by aligning the ribosome with the start codon. So in other words, it's a dock. It's a dock for a biological piece of biological machinery to begin its work on RNA to create that protein. And so now that you have that in your hands, you think, well, now I know what this dock looks like. If I find this, I'm going to find my gene. It's like it's going to come up very, very soon. And so these are what they call genetic landmarks, where if you find this, then you're going to find your gene. And so a lot of the algorithms, the reason why we're talking about this is because a lot of the algorithms that we use to determine where a gene would be is actually found in the, a lot of these, they actually use this kind of information here, these landmarks, to predict the occurrence of genes. And we'll see that toward the end of this class today, what I mean by that, but there are actual systems that automatically annotate DNA sequences. In other words, they look at DNA sequences and they say, I'm guessing that there's this region here is a coding region, and this other region here is not a coding region. And you might ask yourself, well, how does it know that? Because of stuff like this, because of these genetic landmarks. It hasn't actually tested this thing. It's a prediction, after all. It's just predicting. It's saying, I found this genetic landmark, and I know that the ribosome will attach around here, and I know that some other things are going to will happen here, but I know that protein synthesis is going to follow this, in this area very, very, very soon, and so that must be a gene. Interestingly, though, this Shine-Delgarno sequence, if you find this inside the gene, it acts as a kind of a pausing station, I guess, if you will, so that it can somehow, I guess you can use that to regulate how quickly this gene is actually being produced. You may find, for instance, that you're making this, or the cell is making too much protein too quickly, and the protein can't be dealt with in some sense, for whatever reason. But if you have these things in the tend to kind of slow things down. So they're not just landmarks that say that a protein is going to come up, but they actually do something to create protein. So where this is all going is that we're using the familiar to find the unfamiliar. In other words, we're using these Shine-Delgarno sequences, which you can see right in the middle here. That's your Shine-Delgarno, and then you know that gene A has to follow. Now, the trouble is that you don't know what gene A is. You have no idea what that protein is. You have no idea. It's just some protein that comes out. Something is being created in there. But you know that, if you keep searching after the cell of the Shine-Delgarno, you'll find your ATG, which is your start, and then you'll find your TAG codon, which is the end. And so that suggests that you have your start and your end of your gene. And so, again, you don't know what it does, but you know that that is a gene. It's kind of like grabbing a piece of source code from online, where it's like a bunch of source code that you really don't understand. But you recognize that there is a syntax. You recognize that this is Java code, but I don't know what it does. I recognize that it has the curly brackets, it has this, it has that, it has your declarations of variables. But again, I don't know what this thing is. I guess another way of looking at this is like if you look at the node SJ. If you're using JavaScript online, you can download these compressed JavaScript helper files like Node.sj and some other things. It's just like a mess of code. It's all compressed. It's not human readable at all. The computer can read it, but we can't. And so you're looking at all these functions which have these very small letter variables, and so you have no idea what any of this stuff does. But you know that it's Java, or JavaScript rather, because you recognize parts of it, and it's the same way in this when you're looking at code for genes. You recognize there's a certain beginning and an end, and there's this landmark of the Shine-Delgarno, but inside the sequence you have no idea. So that's more research that you may have to do to actually find out what is actually coded by this gene. So we use prediction algorithms that actually do this work for us. I mean, you can't be expected to go through, like, with your own eyes and look at all the sequence and try and recognize these Shine-Dalgarno sequence areas, and there's other ones too, which I have a list later on the slides. But you have all these, I mean, you just can't know these things. But so you have a machine do this for you. There's just so much stuff to look for. And so really what the machine is doing is it's kind of like, it's this kind of problem here where if you look at this text, when you first look at it, it doesn't make any sense at all. But if you somehow rearrange the chunks of words or the chunks of characters, and you kind of figure out where the beginnings and ends are, then eventually you'll figure out where these genes are. And this is really what the algorithms are doing. It seems very low tech when you see kind of like what's happening. But actually, though, the function is itself actually crucial to doing any research. So this is logic will get you from A to B. Imagination will get you everywhere. So you're it's like once you realize like what the beginnings and ends of these things are, then you can start doing some research into the beginnings or into the middle part, and then you can figure out more or less what this thing is supposed to do. So an algorithm has to work with this kind of problem here, where it has to realize that, okay, here's a capital L, that must mean it's the beginning. Here's a period, that must mean this is the end. So this must be a sentence. And so then it will work out what that sentence is by rearranging these letters. And then down here, it says, okay, well, here's an I, which is capitalized, so that must be the beginning of a next sentence. And here is the end of the sentence because there's a period. Now, in terms of like landmarks, we know in the English language that capital letters are landmarks to suggest that this is the beginning of a new sentence. And we know that periods are landmarks to suggest that this is the end of a sentence. Well, I mean, it has other functions too, but in this case, we say that it's the end of the sentence. So, prediction algorithms. Excuse me. So what these prediction algorithms are doing is they're looking at chunks of DNA and they're trying to figure out what's inside that DNA, like what genes does it have here. And also we call those features, because it's not just genes here, but features could also be like, oh, that's interesting. In this DNA we have a Shine-Dalgarno sequence, or here we have another region here that codes for some piece of protein, or here we have some. It's things that kind of punctuate that gene. So what we're doing is we're using alignment again, and I'll keep saying this, that alignment is, I would say, one of the most basic forms of analysis that you'll find in bioinformatics. Seemingly everything is aligned. And here you're probably using something like local alignment, where if you take a piece of code, you use local alignment and you find out what pieces are common to this piece of code that you have here, which are also common to someplace else. In other words, could those common things that I find between this unknown sequence and the other sequence that we have on record in our database, could those common regions, those local common regions, be genes? And if you find out that they are almost entirely the same code, it's very likely that they are conserved portions of coding material. And so you'd use something like BLAST to do that work for you. Again, BLAST is being used. It has so many different functions. But in this case, BLAST would find those common regions and say, oh, you know what, I found what I think is a gene. You look at the other organism for which you know that this is a gene, and then you get an idea about what that gene name is in the other organism, and that gives you an idea about what type of protein is being produced, and then you can give it a name, and you can determine what the function is. And so really, just by doing a basic alignment over this unknown sequence and finding common sequence or common subsequences in here with something else, just because we find that information, now we can make an annotation. That means we know about the content inside this gene. Anyway, there's one thing that I should mention though, the quality versus quantity. BLAST works by something called the heuristic-based model. And what that means is heuristic-based models are different from what they call exhaustive models, in that exhaustive models are every single possible solution is found. In other words, if I have a sequence that I'm comparing to everything that's known, an unknown sequence that I'm comparing to everything that's known, that means that if I use an exhaustive model, then my analysis result would be every single known
gene where this thing is found. That could be an exhaustive list. I mean, I have everything. Then I just have to order my list, and I can just take the thing off the top, and that's like, you know, the closest match to everything that's known. But there's something else called heuristic, where it's faster, and heuristic is saying this is not the best match, but it's pretty good, and it should give you enough information to make an assessment. So heuristic is not exhaustive, but rather it's giving you a pretty good matchup. It's giving you good information from your alignment, which isn't the best necessarily, but it is good enough to work with. And it's for that reason, actually, about heuristics and things, that you got, where was it? Getting back to the our activity here. Here. So it's because of that heuristic that you may have got a different number here. 7,989. I got 7,982, but maybe you got something different. And that's because BLAST was using a heuristic where it said, okay, well, this is close enough. I mean, this should help you along. You should. I can't give you the best, but this is good enough. This result is good enough, and so it should give you enough information to make an assessment. And so prediction algorithms tend to use these heuristics where they kind of like put things together and they don't have the time to do all the deep comparisons, but they do enough. And what I'm saying about these heuristics, though, is that going back to these slides here with the landmarks, a heuristic. We used heuristics just now when we were looking at these different pictures, where I say that this picture here was obviously taken in Paris because of the Eiffel Tower. Never mind the trees, never mind the water, never mind the bridge, never mind the flags in the background, never mind any of that stuff. We didn't bother comparing that at all, but rather we spent our time looking at one thing, which was this Eiffel Tower, and that was very telling. Over here, we looked at Big Ben, and that was telling of that this was taken in London. This was a heuristic because we didn't actually look at every single piece of available information from this photo, but rather we just looked at one specific thing, which was so telling that it told us that it had to be this. It had to be from Paris, that picture, or this other picture had to be from London. And so that is a heuristic approach to things. And so BLAST does the same thing where it will look at these sequences and it says, okay, well, this sequence has to be from this, or has to be from this origin, because I haven't analyzed everything, but the stuff that I did analyze, though, is very telling that it must have come from this. And so you might ask yourself, well, what exactly does it look for then? Well, it looks for like content-based, the content-based analysis here, where it's like looking at what is inside, like what types of code are we looking at here? Does that code look like it could be something that would create protein? There's something else called the nucleotide frequency, and what that means is there are specific types of words that you'd find. Well, for instance, like the Shine-Delcarno word, certain types of words which are rare, but when they show up in the sequence, it means that they do something. Like, for instance, the word in English, if I'm thinking of a word, when I say the word elephant, that only has one meaning to us. It's an animal, a big elephant. It when I say the word elephant, we all know that it means an animal. I wouldn't use that word to describe anything other than discussing a giant animal. I wouldn't say the day was very elephant outside, because that doesn't make any sense. But when I say I saw an elephant, then you know that I'm talking about this giant animal. And so based upon word frequency, you know that every time you hear that word elephant, you know that that has something to do with the discussion of this giant, mammal. So they have words that also show up in DNA that are the same way. Here we have the codon frequency and codon bias. This is actually rather interesting too. Again, when you're making proteins, as we remember, certain types of proteins prefer specific types of makeup of how they're, like specific types of RNA makeups of how they're being created. In other words, we have many different ways of creating a specific amino acid, and different types of proteins prefer different makeups of those codons. Like I think that leucine has like 6 different, and serine perhaps, I think they have like 5 or 6 different DNA or RNA triplets, which can be used to define the amino acid leucine or serine. And so you'll find that different types of proteins prefer different codons to create those proteins. If that makes any sense. Different organisms have different predilections, if you will, for codons. And so you'll find that maybe the mammal types of organisms tend to use these kinds of ways of making their serine, whereas maybe amphibians use a different kind of codon system to make their methionines, or not their leucines or their serines. Their amino acids are created differently. Same code, different code, but same amino acid. Also, you can look at what's called the GC content. That's really crazy, but it's like as you go through the DNA, how many G's and C's are you encountering? And they're saying that where genes are being created, you're likely to find that there are more G's and C's in the DNA at those particular regions. How can we explain that? I think we've already explained this, that G's and C's have 3 bonds, whereas A's and T's have 2 bonds. And so maybe if you have some kind of piece of genetic machinery that's attaching to DNA or rather RNA, and it's doing some things to create proteins, like maybe your ribosome is attaching to your RNA, maybe you need extra strength in your sequence, otherwise the sequence will just break. And so evolution has favored GC content at that region. And so you can look for like elevated GC content, and that might say that this is a gene. But one thing I'll say, though, is that all this stuff that I'm looking at here, all these pieces, while they're all quite, they might seem trivial, but when you start putting them together, you're basically creating a basis of evidence to suggest that your gene is coming up, and this is a gene area. Now, one thing I will say, though, I'm not going to spend too much time talking about this actual this network here. The idea that it's not just this stuff here when you're working when you're trying to predict your. It's not just this stuff here, but it's actually a bunch of other things too that these prediction algorithms do. So, for instance, they take in information from your from the DNA, obviously, but then they compare that DNA information with other pieces of known information about that DNA. They try and figure out like what the proteins could be that are being created from that DNA based upon the information from other proteins which are created from other DNA. And you have a whole series of like events that come together, which is called the annotation pipeline. And these all these pieces of information come together in the algorithm's thinking, in the algorithm's logic, to create basically a probabilistic type of model saying, I've run all these different tests, and it must be this. It must be that this is a gene. And so each time you have a paper that comes out that talks about something like the shell or the Shine-Dalgarno sequence, or there's like something else that's similar, or you find some other landmark, all that information is poured right into this annotation pipeline, which is used by some of these really smart algorithms so that they can determine like what's going on. And interestingly enough, though, when I say what's going on, like what the sequences are doing, what the annotations are, what the proteins are doing. But interesting though, look, BLAST is right here. In fact, BLAST is a very prominent piece of this whole system. So it's funny actually that BLAST is like, if every method were a kind of a rock star, I think that the BLAST method would be the super rock star. Here's another kind of look at this pipeline, the annotation pipeline, where you're looking at you have different kinds of information that pour into this. And this is like how the algorithm would think. This is how the algorithm determines like what the annotations are. So, for instance, you'll have information from proteins, information from genomes, information from the RNA, all this information coming in, which is then basically used to compare to the stuff that the unknown sequence that you're working with, to determine some kind of basically this final sequence, which is the annotation of where you're creating your annotations from. But you have a whole bunch of stuff going on in here. For instance, you have a protein cluster, a protein cluster branch, which then goes to prospline, which is basically looking at the types of looking at what's the probability of having proteins showing up in this area. Then you're looking at frame shift detection here, where frame shift detection is kind of like, for instance, back here, where you're trying to make sense of, well, if I just kind of rearrange the words, can I read a protein out of this whole thing? This is computationally intensive, by the way, because you have to do a whole bunch of rearranging and then read the word and then rearrange it again and read the word, rearrange it, and you have to keep doing that to determine something. And so you have that part here. And so then that's if you're working with proteins. If you're working with genomes, for instance, if you have just DNA, you might convert that DNA into RNA, run some other tests, and then try to determine whether those tests are actually going to produce something that looks like it might create a protein. In other words, if you take your DNA, convert it to RNA, then you create, electronically you create a protein, you check that protein sequence for any other sequences which are similar that you may have found in nature. And if you find that you're getting a sequence that looks like it could have the same kind of syntax to create a protein, then maybe you found a protein. Maybe if you go backwards, you'll find that that's actually a gene. And so on. And so you have all these other ways of, and similar methods here for RNA, but you have all these different ways of kind of testing. They're all a series of small tests that basically build up this body of evidence to say, I believe this is a protein. I've never actually seen this DNA create this protein, but I believe that a protein is here. And that's where this is all coming from. This is another more extensive pipeline here, where you have specific kind of terms to look at here, where they call there's a masking stage, where you're trying to identify, let's read this, trying to identify and ignore non-coding regions. In other words, you're determining what the exons and introns are. I need to know, for instance, what's the coding, what are the pieces of DNA that actually do something? What's the DNA that doesn't seem to do anything in terms of protein making? And so you have to go through this masking stage, which is again done through these algorithms. And as I go through these algorithms, you'll see that they get increasingly complicated so that you can build this body of evidence. Then we have alignment-based predictions. That's again using BLAST. This is what NCBI uses. Then we have the sequence content-based predictions from alignment. What that means is you're again looking at the probability that the sequence that we have in the unknowns, the subsequence in our sequence is similar enough to something else that we've already seen to suggest that that's actually a direct copy of some gene. And then we have, for instance, some more probability-based methods that are added with the results of each one of these, the pink, the blue, the green, and the orange colors here. After each one of these tests has been done, we have to then kind of weigh the results, and we think, okay, what's the probability that we'd find this kind of thing happening in nature? Could it be that this is all just random, or is there something here? And again, once you've run through this whole series of tests, you eventually have some reason, but it's still based in probability, but you have reason to believe that actually this is a protein, or it's not a protein. It's like all this stuff over here. Once we've decided this probability, we can say, well, I'm going to say this is a gene, so therefore I'm going to figure out what kind of gene it is, and then I'm going to go ahead and format my data accordingly. But you'll notice though that if you go back to some of the proteins, you'll find that, for instance, some of the gene records that we've been looking at have been modified recently, and that is because maybe maybe there's a new paper that's connected to that protein. We'll see that in a bit, but maybe somebody has found out that actually, you know what, the protein that we thought it was is not the protein that it is. We find out that actually it's a similar protein, but it's not exactly the same. It has a mutation such that it doesn't do this, it does something else instead, but it's an allele. It's the same idea. And so then they would write a paper, and then that paper would then be kind of put into the record of proteins. This is just another thing here. I discussed this here just now, but basically at the end of this annotation, you have a kind of an idea that you're working with a protein. One thing these proteins or these algorithms do, which I find very interesting, is that they're able to use they use inference here. And so when you're combining or when you're looking for genes, you might look for common regions of code which you've seen somewhere else. So, for example, if you use the word love in Chinese, the word is I. Is that right? I think it's I. I'm hoping I'm pronouncing this correctly. And if you look at Japanese, it's the same. It's the same kind of symbol, if you will. It's not exactly. I mean, it's obviously a different pronunciation, but it's the same kind of thing that you can see that to the uninitiated. And I don't speak Japanese or Chinese, but you can see that there are certain commonalities that exist between the Chinese and the Japanese symbol. There are common regions. I would say that, perhaps this piece on the bottom, on the very the bottom of each of these characters, you have that could be similar. I see a line that's running over the top, And that's also, you can see that it must have the same origins. And you might be able to say that in Korean, I see some of the same types of features in the writing to suggest that perhaps the word origins are similar. And there must be something, there must be some reason why they're written the way that they are. And I would say that there must be one origin, someplace. If you go back further or far enough, you'll find that maybe that's, that the text broke apart. In fact, if you look at English, like some of the numbers that we use, like 0 through 10, you'll find that some of those are from Arabic. You'll find that the ancients, I think around in ancient, Mesopotamia, around in those areas, they were using characters that resemble today's numbers, and so that's actually kind of interesting. So, but if you look back at the history of these and look at the old scrolls, you'll find out that actually we stole our number system from the ancients in the Middle East. And so there are some very common features that you see in writing, and that also happens in DNA. There's some common pieces in the structures of DNA that happen from distantly related things, distantly related sequences. And so that would suggest that, some evidence to say that it must be very similar. One thing that we have to look out for, though, I just thought I'd bring this in here, is that you have different types of DNA to look at, though. And so another piece of evidence you might need to know is that you have prokaryote and eukaryote genomes. Prokaryotes tend to be kind of like bacterial types of cells, and eukaryote tends to be seemingly everything else. But prokaryotes are more simplistic. I hate to say that any of this stuff is simple, but in terms of like eukaryotic cells, prokaryotes are a little bit more simple, whereas eukaryote cells are more complicated. And so I think that's, you can find that in prokaryotes, you'll find that your DNA is a circular, that means that it's a circular chromosome, which means basically it's a ring of DNA, whereas in your eukaryotes, you have kind of linear. And so it makes a difference, though. But the thing is, though, that even though they are different DNA and they behave differently and they're stored differently in the cell, just going back to this over here, you can find that there's perhaps enough common information to suggest that the syntax was the same. It's arguably the same. Anyway, another thing that's actually interesting in eukaryote cells, though, and some prokaryote cells, actually eukaryote cells, is that you have, for instance, mitochondria and chloroplasts. These are actually links you can click on. But you have these mitochondria and chloroplasts, and these organelles have their own DNA too. And so you're not just looking at like cellular DNA, but you're also looking at the DNA that comes out of chloroplasts and mitochondria. It's absolutely fascinating to see mitochondria, these organelles inside the cell, the eukaryote cell, that are the ATP generators of the cell, the energy producers. Because those organelles have their own DNA, it makes you think that maybe they were their own like bacterial, cells or smaller primitive cells that took up residency in eukaryotes. And there's a lot of evidence for that, but they're also saying that perhaps it isn't that simple. Maybe there's other stuff going on. In other words, maybe, for instance, chloroplasts and mitochondria needed their own DNA to be able to kind of organize themselves to produce the energy that the cell needed without having to go to the cell and asking for permission to create more mitochondria. What I'm trying to say, though, is that if you exercise and spend a lot of time in the gym and do a lot of weights and everything, then you have more mitochondria in your body than somebody who does not. And that means that you're producing more energy producers than somebody who doesn't go to the gym. And that's because you need that energy to be able to lift those weights. And so it's actually kind of interesting. So one of the theories is that maybe you don't need to go back to the cell every time and get more, and get permission to make more mitochondria when you need it, because that just takes time, but rather the mitochondria kind of regulate themselves. And that actually does make sense, though, because mitochondria do regulate a lot of things in the cell. But that's a side story here. But getting back to the thing here, but the reason why we need to know about prokaryotes and eukaryotes is because in prokaryotes you have things like the Schindel-Garno, but then in eukaryotes you have something called the Tata box or the Tata box. And these really serve the same kinds of functions here where they are landmarks. This is some. This is actually kind of interesting here. These are just different types of, just to give you an idea about prokaryote and eukaryote cells. I might skip this for a second here, just in the interest of time here. But these are other kinds of landmarks you'd find in prokaryote and eukaryote cells, where you have the Tata box, and you'd have, like, for instance, the TAA or TATAAT. These are other, like you find that in the prokaryotes, the TATAAT. That's a prokaryote thing, but the Tata box, TATAWAW, that's a that's like a wild card. But it's like you're saying that you have similar kinds of landmarks, but they're not exactly the same. And so that suggests that perhaps we're dealing with something which is like the landmarks are similar enough, but they're not exactly the same. But these are other kinds of landmarks that these algorithms would look for. We have 4 minutes. I think that maybe I'll try and go. I'll go for it and see what we can do. I wanted to show you something today, which is called the Open Reading Frame Viewer, which comes from our very own NCBI. And so hopefully this link, let's go ahead and copy this here. But this link is actually quite exciting because what it does is, this is a place where you can put in a piece of DNA. Imagine you're a researcher and someone gives you this DNA from some unknown place. You enter this into this viewer. This will tell you like what's where are the genes in this, or what kinds of proteins can I expect to get out of this? So if you go back to the slides here, you'll see that, so these are just the links. Bring the tool here. You'll see that, for instance, we're going to look at our old friend here, the cert1 accession, which is this KF881970. If I can remember this, I can put this in here. I'll put this down here in this thing here, KF881970. Am I getting this thing correct? Let me just make sure that I got this right. 881970. I'll need the session number, so you put the session number down here. Then I'm just going to leave all these parameters exactly as they are. I'm not going to worry about anything. And I hit, but this is interesting, by the way, here. The ORF Open Reading Frame Start Codon. In other words, it's saying, surely you can give me some information about where you think the gene would begin, at ATG or where. But I'll just leave this as it is. I'll hit submit. And what this is going to do is it's going to look at my sequence that this is actually a sequence that we know here. This is our cert sequence. But it's going to go through that sequence, and it's going to try and determine what the landmarks are to determine where the proteins are. So in other words, I mean, this is research. This is well-researched stuff. But maybe this particular gene that I'm looking at right here, though, maybe this didn't have the research, but maybe, for instance, it was studied by the algorithms and found out that this first big red bar is actually a coding region inside the sequence. And so let me just show you something. If I click on, I don't know if you can see where I'm clicking here, but there's this little button, it's like a slide rule at the top here, which actually allows me to zoom in. And so I'm now looking at the code itself. And can I zoom around here? Or maybe I can't zoom around here. How do I, yeah, okay. Well, what I'm saying is, like this over here, you can see this word right here that says ORF1. That is what they call an open reading frame 1, which is a place which in the DNA which is supposedly a gene. It's a protein. And so by having my mouse running over this thing, I can see like all these different areas here that I can go ahead and click on and get some more information about what exactly that protein is. But in this, the most important thing here is that in this sequence, it says that if I look at the sequence in terms of, like, starting from the very first letter or the first character, I can find these proteins. If I look at the second letter or the second character, I can find these particular proteins. Or if I start at the third frame, I can find these other proteins. And so you're actually able to kind of look inside this DNA and find different types of proteins that exist. Yeah, so it's actually quite exciting. And so you have all this power in here. And I need all these slides right here so that you can study this at home if you'd like. But basically, though, you have information about what the embedded proteins could possibly be.